For decades, the biological classification of the Elephantidae family was dictated strictly by geography. Textbooks recorded two distinct species based on their continents of origin, Loxodonta africana in Africa and Elephas maximus in Asia. Within the African continent, biologists routinely observed physical variations among the populations. Elephants living in dense tropical forests exhibited distinct morphological traits, such as rounded ears and thinner, straighter tusks, compared to the larger elephants roaming the open savanna. These physical differences ignited a long-running debate. Some researchers argued these traits were significant enough to warrant categorizing the forest elephant as its own species. Others maintained they were simply localized adaptations, minor superficial variations within a single, continuous African species. The problem with resolving this debate visually is that morphological traits are highly malleable. Physical appearance adapts to local environmental pressures rapidly, making it an unreliable metric for proving reproductive isolation. Mapping the true evolutionary history of the elephant requires looking past the macroscopic scale of tusks and ears, focusing instead on varying resolutions of modern genetic data. The single-species model for African elephants was a decades-long illusion based entirely on human observation. It persisted until the advent of targeted molecular sequencing. Decoding the complete elephant family tree required two entirely different genomic tools, each matched to the specific evolutionary timeline of the African and Asian lineages. The first tool is targeted nuclear locus sampling, which isolates extremely specific segments of DNA, in this case 1,732 base pairs. The second is whole genome sequencing, which maps a colossal 3.4 billion base pairs across an organism's entire genetic code. Because the populations in Africa diverged millions of years ago, targeted nuclear genes were sufficient. The genetic distance between them is wide enough that a small, precise sample can easily detect the deep, ancient split. In Asia, however, the evolutionary timeline is compressed, obscured by recent, severe human-induced habitat fracturing. Untangling these closely related populations required the massive resolution of whole genome sequencing to sift through the noise and identify hidden geographic boundaries. This genomic mapping reorganizes taxonomic classifications and dictates how conservation strategies are applied to highly inbred populations facing severe habitat loss. Aligning the resolution of genetic data to the specific population provides the foundational framework for long-term conservation management. To resolve the African debate, a 2001 study by Roca et al. Extracted DNA via biopsy darts from 195 free-ranging African elephants across 21 populations. They sequenced 1,732 base pairs across 4 distinct nuclear genes. This phylogenetic tree generated from the data reveals a deep bifurcation between the elephants of the tropical forests and the savanna. The split is confirmed by bootstrap support values at the primary nodes, showing high confidence in their separation. When analyzing the variants, the researchers found that the sheer genetic distance, combined with a timeline stretching back to the late Pliocene, mathematically dismantles the theory of a single African species. The data points conclusively to 2 separate species. This minimum spanning network diagram tests for reproductive isolation. Hatch marks highlight 6 fixed nucleotide changes separating the African forest and savanna clusters. This evolutionary gap is nearly as large as the gap separating either African group from the Asian elephant cluster. The genetic mapping correlates perfectly with field findings. Despite overlapping physical ranges in Central Africa, researchers found 0 F1 hybrid genotypes across all sampled populations. A complete absence of first-generation hybrids proves there is no active gene flow between these groups. They are functionally and reproductively isolated in the wild. The combined weight of deep genetic distance and strict reproductive isolation leaves no room for debate. It confirms the formal elevation of the African forest elephant to a distinct species, Loxodonta cyclodes, standing alongside the savanna elephant, Loxodonta africana. While the African lineage diverged millions of years ago, the Asian elephant presents a different taxonomic challenge. Its evolutionary history has been heavily obscured by recent human interference and severe habitat fragmentation. To resolve this, a 2025 study by Kappelhoff et al. Utilized whole genome sequencing. They mapped the complete genomes of 27 wild-born Asian elephants at roughly 25 times coverage. This map shows the current geographic distribution of the surviving Asian populations. They exist in highly fragmented, isolated pockets across South and Southeast Asia, historically categorized into 4 main subspecies. This admixture bar chart helps researchers determine if these geographically isolated pockets are genetically distinct. Looking strictly at the K=4 row, strict population boundaries emerge. We see solid, unadmixed color blocks for the elephants of Borneo, Sumatra, Sri Lanka, and the Asian mainland. An unadmixed block indicates that these populations have been cut off from gene flow for significant periods of evolutionary time. There is no modern genetic crossover between these groups. Whole genome sequencing proves the Asian elephant is not a single homogenized population. It is a collection of rigidly structured, genetically isolated geographic lineages. This genomic precision also resolved a long-standing historical myth. For decades, it was heavily debated whether the elephants on Borneo were native or simply the descendants of a domesticated herd introduced by humans in the 16th century. This is a principal component analysis plot measuring genetic drift. The red cluster representing the Bornean elephants is isolated at the extreme edge of the chart, highly divergent from all other Asian populations. By utilizing a molecular clock analysis of orthologous genes, researchers decisively dated this divergence. Using a molecular clock analysis, researchers dated this divergence. The Bornean elephant split from Sumatran lineages 170,000 years ago, deep in the Pleistocene epoch. The data also explains why the 16th century introduction myth existed in the first place. The timeline graph shows a sudden, precipitous drop in the Bornean elephant's effective population size, creating catastrophic levels of recent inbreeding. An analysis of the runs of homozygosity across their genome reveals a severe population bottleneck that occurred just 8 to 38 generations ago. To early observers analyzing limited data, the genetic signature of this massive population collapse mimicked the profile of a small introduced founder herder. The sequencing resolves the debate entirely. Bornean elephants are a deeply diverged, endemic lineage that survived the Pleistocene, only to suffer a catastrophic collapse. The reliance on outward physical traits led to a flawed understanding of Proboscidean taxonomy. That model must be completely discarded in favor of a strictly genomic family tree. Rebuilding the tree starts at the basal root, splitting into Loxodonta and Elephas. 2.6 million years ago, the left branch splits into distinct savanna and forest species. On the right branch, 170,000 years ago, geographic fracturing created today's discrete Asian subspecies. The genomic evidence establishes 3 deeply diverged species existing alongside highly structured, genetically isolated Asian lineages. Managing these uniquely adapted lineages as interchangeable subjects in conservation programs carries severe, irreversible consequences. Crossing deeply diverged genetics triggers outbreeding depression. It actively destroys the localized biological adaptations that each lineage spent hundreds of thousands of years evolving. The genetic data categorizes the Sumatran, Bornean, and Sri Lankan elephants specifically as evolutionary significant units. To survive, they require entirely isolated management. These units are highly fragile. Historical bottlenecks left them with a high genetic load, possessing unique vulnerabilities hidden deep within their genetic code. Both captive breeding programs and wild habitat protections must shift their operational models immediately. The mandate is to manage specific, isolated genomes, rather than generic physical phenotypes. Preserving the elephant family requires protecting the invisible genetic architectures that allowed these lineages to survive the Pleistocene, architectures we finally have technology to see.